Okay, so this is the second part of host defenses, and we will primarily get into and then we'll get into the adaptive immune system. So with your specific immunity, this is what we call acquired specific immunity, and it's called acquired because you have to acquire it over the course of development upon exposure to particular antigens. It is the product of B and T lymphocytes. The B cells and the T cells are what are part of the adaptive immune system. The lymphocytes will undergo a selective process that specializes them for reaching to only one specific antigen or to an immunogen. Immunocompetence, this is the ability of the body to react with countless foreign substances. Now, antigens or immunogens, these are chemical substances that stimulate a response by a T and B cell. It could be a protein, it could be a polysaccharide molecule on or inside the cells, and viruses. Any exposed or released protein or polysaccharide could potentially be an antigen. Usually our own antigens do not evoke a response from our own immune system. That would be bad if your own antigens are able to trigger an immune response. The other way I like to think of antigens are anything that the body perceives as non-self. So let's look at some of the characteristics of specific immunity. Specificity, highly specific to the antigen against which the third line of defense is directed. The other nice thing is that you have memory. This memory is the ability to rapidly mobilize lymphocytes that have been programmed to recall their first engagement with the invader and rush to attack again. Part of the adaptive immune response is the first time you're exposed to an antigen, it's oftentimes a very slow and very weak response, but you end up producing memory so that if you see that particular antigen again, you get a very rapid and immediate response. This idea of memory here is really what allows for vaccines to be as effective as they are. So looking at specific immunity, what makes it special? One is the specificity. The specificity is the response focused on a single antigen. Diversity. You always have at least one cell that can react against any antigen. Inducibility, it only gets turned on when it's triggered. Clonality, this generates millions of cells with the same specific immunity. Tolerance does not react with self-antigens. Again, that would be bad. And then memory, the ability to recall when you see that particular antigen again. So looking at the principal stages of immunological development and interaction, lymphocyte development and differentiation will be the first. Then we have the presentation of antigens. And then you have the antigen challenge of either the B or T lymphocytes. They're going to give different responses. B lymphocyte response is going to produce the activities of antibodies. And essentially, what you're getting at the end of the day with B cell response is antibody production. T lymphocyte response is going to be cell-mediated immunity, which will attack very specific cells. Now, markers on surfaces of cells are involved in recognition of self and non-self. And this is an important concept in the immune system. It's the ability to recognize self, which is your particular cells, versus non-self, which would be anything that is not you. Major functions of the immune system markers is attachment to non-self or foreign antigens, the binding to cell surface receptors that indicate self, and your self antigens that we have on human cells are called MHC receptors. Also receiving and transmitting chemical messengers to coordinate the response and aiding in cellular development. Now there are three classes of MHC genes. Class 1 MHC genes or markers are going to appear on all nucleated cells. They display unique characteristics of self. They allow for recognition of self and the regulation of the immune reactions. And each human inherits a particular combination of class 1 MHC genes. Class 2 MHC genes, these code for immune regulatory markers found on macrophages, dendritic cells, and B cells. They are involved in presenting antigens to T cells during cooperative immune reactions. And then the class 3 MHC genes encode proteins involved with the complement system. And here you can see these MHC genes. They are essentially receptors on the surface of our immune cells. We also have CD molecules, or cluster of differentiation molecules. That's what CD stands for. These are markers important in immunity. They are found on the membranes of a variety of different cells involved in the immune response. And there have been over 400 different CD molecules that have been identified. So looking at the surface of T cells and B cells, what we see is we can have these MHC markers, as you can see there, receptors. You can also have antigen binding sites, which are going to be the receptors to bind to very specific antigens. And then you can also have some of these CD molecules present on the surface. Okay, so entrance and presentation of antigens. How do antigens get presented to the immune system? You have tissue macrophages. These ingest pathogens and induce inflammatory response in the tissues if appropriate. Again, this is more of an innate response, but it's going to trigger up the presentation of antigens. Tissue dendritic cells, this will ingest the antigen. It will migrate to the nearest lymphoid organ, and it will process and present the antigen to the T lymphocytes. You'll then have your antigen presenting cells. These are dendritic cells, macrophages, and B cells. The B and T cells will proliferate and differentiate when challenged by an antigen. And when they proliferate, you're essentially going to form a clone. A clone is a genetically identical cell, some of which are memory cells. So how do T cells respond to an antigen? There are three main functional types of T cells. You have helper T cells, which will activate macrophages, assist B cell processes, and help to activate cytotoxic T cells. Helper T cells really are the primary controller of our immune response. HIV does attack our helper T cells. The regulatory T cells, they control the T cell response, and the cytotoxic T cells lead to the destruction of infected host cells and other foreign cells. When activated by an antigen, a B cell will divide, giving rise to what we call plasma cells. Plasma cells have the same reactive profile as the B cell that it originated from. However, plasma cells essentially produce and release antibodies into the tissue and blood. Antibodies will then attach to the antigen for which they are specific and mark it for destruction or neutralization. So looking at the markers that are found on T cells, we have antigen-specific T cell receptors. You also have CD3 markers. These surround the T cell receptor and they assist in the binding of the T cell receptor. And then there's two called CD4 and CD8. CD4 will bind to MHC class 2 molecules. CD8s are found on the cytotoxic T cells and bind to MHC class 1 molecules. So essentially your CD4s are your helper T cells, CD8s are your cytotoxic T cells. Now T cells will constantly circulate between the lymphatic and general circulatory system. They migrate to specific T cell areas of the lymph nodes and spleen. There are estimated that there are over 1 billion T cells that pass through the lymphatic and general circulation per day. B cells, on the other hand, you have hundreds of millions of distinct B cells develop as a result of gene modification and selection. Circulation of B cells through the blood, homing to specific sites in the lymph nodes, spleen, and other lymphoid tissues where they take up residence. B cells will have immunoglobulins as surface receptors. And again, their main job is to look for foreign antigens and produce antibodies if they should become activated. So here we can see contrasting properties of B cells and T cells. This is a nice table for you guys to look at the key differences between these two cell types. So each naive lymphocyte bears an antigen receptor that recognizes a unique antigen. Stem cells become granulocytes, monocytes, or lymphocytes in the bone marrow. Lymphocytes become either B or T cells. B cells stay in the bone marrow. T cells migrate to the thymus. Each builds a unique antigen receptor. Both B and T cells migrate to secondary lymphoid tissues. By the time B and T cells reach the lymphoid tissues, each one is equipped to respond to a single unique antigen. And here we can see the production of the B and T cells in the bone marrow, their maturation in either the bone marrow or the thymus, and then their release to circulation. Essentially, the stage is described right here. Okay, sorry. So the mechanisms behind antibody variability. We have genes that encode variable regions. What happens is those particular genes, depending on the introns and exons of those genes, you end up with different RNA transcripts from the same gene. The different RNA transcripts produce different amino acid sequences, which amino acids make proteins, so you're essentially making different antibody receptors or immunoglobulin receptors. The specific B cell receptor or immunoglobulin is composed of 2 heavy chains and 2 light chains. One light chain is bound to one heavy chain. The 2 heavy chains are bound to each other with disulfide bonds, and they create a symmetrical Y-shaped arrangement, as you can see here. The antigen binding sites, these are pockets at the ends of the forks that are formed by the light and heavy chains. They can be highly variable in shape and fit a wide range of antigens. The variable regions, these are found in the antigen binding sites. The amino acid position is highly varied from one clone of B lymphocytes to another as a result of genetic reassortment. And the constant regions, these are the amino acids that do not vary from one receptor to another. Remember, these receptors have to be different because they're picking up many, many different specific types of antigens. So essentially, you're going to have lots of B cells roaming around that all have very different variable regions on their B cell receptors. The T cell receptors, these are similar to the B cell receptors. They're formed by genetic modifications. They have variable and constant regions. They are inserted into the membrane, and it has antigen binding site formed from 2 parallel polypeptide chains. Unlike the immunoglobulins, these are much more smaller, and they're never secreted. Okay, so clonal selection theory of lymphocyte development and diversity. So each genetically unique line of lymphocytes arises from extensive recombinations of surface proteins termed a clone. This proliferative stage of lymphocyte development does not require the actual presence of a foreign antigen. At the same time, any lymphocytes that develop a specificity for self molecules could be harmful, meaning that if they are forming a receptor that would potentially react to a self antigen, that would be harmful, and you need to get rid of those. Okay, this process of getting rid of these ones that could be harmful to you is called clonal deletion, and this leads to immune tolerance. Now what you see down here is the specificity for a single epitope is programmed into the lymphocyte and is set for the life of the given cell. The end result is an enormous pool of mature but naive lymphocytes that are ready to further differentiate under the influence of certain organs and immune stimuli. And if you look, the receptors that were formed, they're all different. Okay, they're all here, or they're all the same lymphocyte, but they're all different. When any epitope enters the immune surveillance system, it encounters specific lymphocytes ready to recognize it. Okay. Such contact will stimulate the activation of the clone, leading to genetic changes that cause it to differentiate into an effector cell, and then you get rapid mitotic division. So here we have an epitope or an antigen. It binds to this one very specific clone right here, and this will actually undergo mitosis to start to replicate. So immune tolerance, this is tolerance to self. It's the removal of any potentially harmful clones through clonal deletion. Some autoimmune diseases are thought to be caused by the loss of immune tolerance, the survival of the forbidden clones, or failure of other systems. Now let's talk about how the antigens get presented. An antigen, this is a substance that provokes an immune response in specific lymphocytes. To be perceived as an antigen or immunogen, a substance must meet certain requirements in foreignness, shape, size, and accessibility. Antigenicity, this is the property of behaving as an antigen. Antigens must be perceived as foreign. Whole microbes or parts of microbes can be considered antigens. Cells are substances that arise from other humans, animals, plants, or various molecules. For example, getting the wrong type of blood or a tissue transplant from another person. Molecules of complex composition, such as proteins or protein-containing compounds, are more immunogenic than repetitive polymers composed of a single unit. There are various categories of antigens. You can have proteins and polypeptides, lipoproteins, which are more of a lipid protein base, glycoproteins, more of a sugar protein base, nucleoproteins, more of a nucleic acid protein base. Certain polysaccharides and lipopolysaccharides can also act as antigens. A substance must be large enough to catch the attention of the surveillance cells to initiate an immune response. Generally, large antigens are much better off than smaller ones. Molecules with a molecular weight of less than 1,000 are seldom complete antigens. From 1,000 to about 10,000 molecular weight, they are considered weak antigens. Anything above 100 is considered a strong antigen. Epitopes, these are a portion of the antigen molecule that is recognized and responded to by a lymphocyte. The primary signal that the molecule is actually foreign. Haptens, these consist only of determinant groups. They are too small by themselves to elicit an immune response. If linked to a carrier group, the combined molecule can then develop immunogenicity. So haptens sort of have to come together and sort of form a complex in order for it to be immunogenic. Superantigens, these are bacterial toxins. They are potent stimuli for T cells. They activate T cells at a rate of 100 times greater than ordinary antigens. They can also result in overwhelming release of cytokines and cell death. Toxic shock syndrome and certain autoimmune diseases are associated with these types of antigens. So looking at the role of antigen processing and presentation, antigens must be further acted upon and formally presented to the antigen presenting cells. Your antigen presenting cells will include your macrophages, B cells, and dendritic cells. What happens is the antigen presenting cells will engulf a microbe. It will take it into intracellular vesicles. It will then degrade it into smaller peptides or pieces. The antigen pieces are then complex with the MHC2 receptors, and they are transported to the antigen presenting cell's membrane, as shown right here. From this surface location, the antigens are then presented to a helper T cell, which is specific for the antigen being presented. Here we can see a particular dendritic cell, which is a macrophagocytic cell, engulfing a fungal spore. So as the antigen enters with the antigen presenting cell and activates the CD4 and CD8 cells, you get this reaction here. So when the helper T cells are stimulated by the antigen MHC complex, they then differentiate into helper T cell 1, helper T cell 2, and helper T cell 17, and other types of T cells. What then happens is these T cells will then go on to activate various other aspects, whether it be activation of B cells, which will start to produce plasma cells and then produce antibodies. That's the ultimate response here. Some of these other ones can end up stimulating macrophages, and then you can also stimulate things like inflammation or other responses of the immune system. For T cell activation, it's very similar. Here you have the antigen presenting cell will bind to the helper T cell. This will activate the cytotoxic T cell. The cytotoxic T cell will then go to the infected cell and cause that infected cell to be destroyed. A compromised cell or infected host cell would be one that's infected with a virus or a cancer cell. And T cells kill your cells that are compromised. So these CD8 T cells will kill either a virus infected cell or a cancer cell. And essentially a big avenue of cancer therapy right now is to make your T cells smarter. Cancer therapy, one of the major avenues of treatment is what we call immunotherapy, this is exactly what this is doing here. Okay. And here are the characteristics of the different types of T cells and their functions. So the events in B cell activation and antibody synthesis, the first thing that has to happen is you have to bind the antigen. So you already have a pre-committed B cell that binds only to the particular antigen that fits that receptor. Antigen presenting cells can also present the antigens to the naive B cell. The second stage is antigen has to be processed and presented. The antigen is endocytosed by the B cell. It is degraded into smaller pieces. The antigen is then bound to the MH2 receptors on the surface of the B cell. Once that occurs, the B cell will then present that antigen to a helper T cell. For most cells to become functional, they must interact with a helper T cell that bears the receptors for the antigen from the same microbe. This T cell also has to have been activated by the antigen presenting cell. And then what you end up getting is if the helper T cell agrees that this is a foreign antigen, the B cell will then get activated. The combination of these stimuli on the membrane receptors causes a signal to be transmitted internally to the B cell nucleus. These events will then trigger B cell activation. Once the B cell is activated, you'll then go through to differentiation. The original activated B cell now differentiates into three types of advanced B cells, that of plasma cells, memory cells, and regulatory cells. The clonal selection is when each of the three cells will expand its populations, making clones, by undergoing numerous mitotic divisions. The plasma cells will then go to produce lots of antibodies. The memory cells will just stay in the population and they will stay there pretty much for life. So cell-mediated immunity, this requires a direct involvement of T lymphocytes throughout the course of the reaction. Among the most complex and diverse in the immune system, it's involved in several subsets of T cells whose actions are dictated by the antigen-presenting cells that activate them. The T cells require some type of MHC recognition before they can be activated. All produce cytokines with the spectrum of biological effects. Your helper T cells play a central role in regulating immune reactions to the antigens, including the regulation of B cells and other T cells. They are involved in activating macrophages directly by receptor contact or indirectly by releasing cytokines. Stimulation of the helper T cells by the antigen MHC complex differentiates them into several different types, as we talked about, depending on what type of cytokines the antigen presenting cell also presents. Your regulatory T cells, they maintain that happy medium because you don't want your immune system just to get out of control, because that can actually be detrimental to you as well. The regulatory T cells in the helper T cell class carry CD4 markers. They control the inflammatory process, they prevent autoimmunity, and they help to make sure the immune response does not inappropriately target your own normal microbiome. The B cells become activated to become plasma cells by cytokines from the activated T cells. Already activated regulatory B cells can secrete their own cytokines, which will help to dampen the T cell response. Your cytotoxic T cells, these are the cells that kill. Cytotoxicity is the capacity of certain T cells to kill specific target cells. The killer T cell or CD8 activation must recognize a foreign peptide complex with self-MHC1 presented to it and mount a direct attack on the target cell. There are several cytotoxic T cell secretions after activation. The first is perforins. These are proteins that punch holes into the membranes of your target cells. They cause ions to leak out, essentially going to cause that cell to lyse. Granzymes. These are enzymes that will attack the proteins of the target cells. After they attack with the perforins and granzymes, the target cells typically will go through death through a process called apoptosis. And here we can see cytotoxic T cells actually destroying a virus-infected cell or cancer cells, okay? So again, B cells and antibody production is going to be for foreign cells, such as bacteria, fungal spores, parasites, things like that. Cytotoxic T cell response is going to kill your cells that are virally infected, that are cancer cells, or actually cells from other humans. Gamma delta T cells, these are distinct from other T cells. They have T cell receptors rearranged to recognize a wide range of antigens. They respond to certain PAMPs on microorganisms. They respond very quickly, and they help to produce memory cells when they are activated. Natural killer cells, these are lymphocytes. Related to T cells, they lack specificity for antigens. They can circulate through the spleen, blood, and lungs. They're the first killer cells to attack cancer cells and virus-infected cells in a similar manner to T cells. They are not considered part of the specific cell-mediated immunity because they do not possess antigen receptors. Natural killer T cells, this is a hybrid type. They display T cell receptors as well as natural killer cell markers. They are assimilated by glycolipids on foreign cells, rapidly produce cytokines, granzymes, and perforins. They trigger self-destruction into the target cells. Okay, and those are all the different types of T cells that we have explained. Again, they're all summarized on that table that we saw prior. Okay, so immunoglobulins, there are four different, immunoglobulins. This is your typical antibody structure, okay? Much like the receptors, we have two functionally distinct fragments. You have the antigen binding fragments. This is their variable regions. And then you have the crystallizable fragments, which is the rest of the molecule. So the variable regions will consist of heavy and light chains. They are a special region that attaches essentially the specific antigens. The FC fragment, this will serve as an anchor, and it's involved in the binding to various cells and molecules of the immune system. So antigen-antibody interactions and the functions of the FAB segments. The hypervariable region, this is a site on the antibody where the epitope binds. Amino acid content is extremely varied, and a minimal complementary fit is necessary for the antigen to be held effectively. So what are the functions of antibodies? What do they do? Why are they important? Well, first, antibodies can coat the surface of a bacterium, preventing its normal function and reproduction in various ways. For example, when antibodies bind to microbes, the antibodies will then encourage the uptake of the microbe by a phagocytic cell, essentially by flagging them. Picture this as like little red flags now all around this thing. This process is called opsonization. Opsonization has been linked to putting handles on slippery objects to provide the phagocytes to get a better grip. Neutralization is a second thing that antibodies can do. In neutralization reactions, antibodies fill the surface receptors on the virus or the active site of the microbial enzymes, and it actually prevents them from attaching normally. Agglutination is essentially clumping a bunch of bacteria together, as you can see here, and this actually will cause the macrophages to be able to locate these and engulf them easier. Complement, activation of complement, which will cause the bacterial cells to lyse. And then finally, it could act as an antitoxin. This is a special type of antibody that's produced to neutralize the effects of toxins that get produced. So here the bacteria is producing a toxin, but if the antibodies bind to it, the toxin can no longer bind to what it was going to cause the damage. Okay, so opsonization is the attachment of an antibody to foreign cells and viruses to expose the epitopes to the phagocytic cells. Accessory molecules on the immunoglobulins, you have the J trains. These will join the monomers of certain types of antibodies, IgA and IgM together. Here we have 5 different classes of antibodies. I don't expect you guys to know the difference between all 5, but they are listed here. I would just sort of, just look mainly at the, don't worry about a lot of stuff in the middle here, but just kind of know they're, IgG, this is for long-term immunity. IgA is typically secreted on mucous membranes. So just know generally what each one of these types is responsible for. We can measure the production of antibodies over time. This is called a titer, the concentration of antibodies that's found in blood serum. It can be measured over time to determine how the immune system reacts to an antigen. Some of you may be familiar with this. This is what they're looking at with the vaccine development for COVID, is to seeing how your antibody response is occurring. When we talk about production of antibodies, first exposure will cause a primary response. You typically will have a latent period. This is marked by a lack of antibody production. The antigen is concentrated in lymphoid tissues. It's not really as dispersed. And plasma cells produce antibodies. The serum titer increases to a plateau, and then it tapers off to very low levels over the weeks and months. A secondary response or amnestic response, this occurs when the immune system is exposed again to the same immunogen weeks, months, or even years later. The rate of antibody synthesis, you'll get a peak titer, meaning you'll get a much more robust response, and the length of the antibody response is greatly increased over time as compared to the primary response. Again, vaccines, when they administer a vaccine, they're hoping to get the primary response. So that if you should see the antigen in real life, you would get the secondary response to combat it, hopefully before the antigen is able to even cause any symptoms for you. So this is just a little schematic that describes the primary and secondary responses to antibody production. We have what's called artificial immunity, or artificial passive immunity. This is when they will actually inject antibodies directly into you. The protection typically lasts for about 2 to 3 months. An example would be like if you go for a tetanus shot, they give you the artificial passive immunity because you're exhibiting signs of tetanus already. You don't have time to develop your own immune response. So what they're doing is they're actually injecting you with artificially passive immunity, which means they are injecting you directly with antibody, which will bind up the tetanus toxin so that you don't have the serious side effects of that. That's one example. Okay. Artificial active immunity. This is the process of vaccination. It's called artificial because it's developed in a lab. They're injecting it into you. But it's called active immunity because you're actively producing an immune response to it. So essentially, vaccination is exposing a person to material that is antigenic but not pathogenic. The discovery of vaccination was one of the farthest reaching and most important developments in medical science. And I'm sure many of you, if you're following COVID vaccine development, you're really getting a firsthand approach on how this all works. The basic principles of vaccination is to stimulate a primary immune response that primes the immune system for future exposure to virulent pathogens. If the pathogen later enters the body, the secondary immune response will be immediate, powerful, and sustained. So the principles of vaccine preparation, some considerations is the antigen that's selected, how effective is it, how easy is it to administer, the safety, and the cost. Again, all of these factors are actually currently in the news right now. They've talked about the effectiveness. We have 2 COVID vaccines that have just been released. Data has been released. The vaccines have not yet been released. The data suggests that they are quite effective. They're both about 95% effective. As far as the ease of administration, we know the Pfizer vaccine is a little more complicated because it has to be kept at minus 80 degrees. So that's a little more complicated. Safety is a big one. You don't want the vaccine to be more harmful than the actual issue. And cost. Cost doesn't seem to be too much of an issue here as the government is mostly picking up that tab. But, with typical vaccinations, that is something that you need to consider. Can't be out of reach. So the requirements for an effective vaccine, it should have low-level adverse side effects or toxicity and not cause serious harm. It should protect against exposure to natural, wild forms of the pathogen. It should stimulate both antibody B cell response and cell-mediated T cell responses. It should have long-term lasting effects, the ability to produce memory. It should not require numerous doses or numerous boosters, not to say there may not be boosters, say 5, 10, 20 years. And it should be inexpensive and have relatively long shelf life and be easy to administer. Different types of vaccines. You have whole cell vaccines. This is exactly what it sounds like. They're giving a whole cell to you. Typically, these cells are inactivated or killed. Subunit vaccines. This is not a whole cell. It's maybe taking a few proteins from a bacteria or a few proteins from a virus. But those proteins that they're taking are antigenic, which means they will mount an immune response. The development of new vaccines. Again, your textbook doesn't have COVID vaccine in it because your textbook is already in print. But that is something that you guys can witness, front and center. Dozens of bacterial, viral, protozoan, fungal diseases still remain without functional vaccines, such as malaria. There's no vaccine for HIV. Various diarrheal diseases, various respiratory diseases, and various worm infestations. Most existing vaccines are out of reach for much of the world's population. DNA vaccines. Microbial DNA is inserted into a plasma vector and then inoculated into the recipient. Human cells take up the plasma and express the microbial DNA in the form of a protein. And then what happens is these foreign proteins will be recognized by the immune system and activate a B and T cell response. The routes of administration and side effects of vaccines. Traditional routes of vaccine administration is either subcutaneous, intramuscular, or intradermal. They also have oral or nasal vaccines. are available for a few diseases. Most vaccines will have an adjuvant. This is a special binding substance that's added to some vaccines. Any compound that enhances the immunogenicity or prolongs the antigen. Unfortunately, adjuvants really get your immune system pissed off. They rev it up, which is what you want because you want your immune system to focus on what was just administered. However, when you sort of really rev up the immune system like that, some people can feel like crap. And that's why a lot of times when people get a vaccine, they say, oh, I got sick. No, you didn't get sick. All that happened was that your immune system is actually really pissed off and it's doing its job. And actually, if you feel kind of crappy after a vaccine, you know that the vaccine is doing its job. Some vaccine side effects, local reactions at the injection site, fever, allergies can all arise. Some people have attempted to link childhood vaccines with the development of diabetes, autism, and asthma. Very public debate about vaccine safety occurs. It's difficult for parents and consumers to discriminate between good and bad information, and scientists are trying to address the parent fears and provide reliable information. And by the way, there is no direct evidence, none, between diabetes, autism, and asthma. Herd immunity, this is another term you've heard recently in the news if you're paying attention. Each microorganism requires a certain density of susceptible individuals in a population, which is referred to as the herd, to maintain the chain of transmission. With a sufficient number of immune individuals, the microbe does not spread. Collective immunity through mass immunizations confers indirect protection or on non-immune members. The maintenance of herd immunity through immunization prevents epidemics. So who and when to vaccinate? Vaccination has been traditionally the most prominent in childhood. Adults need vaccinations as well to boost an older immunization to protect against adult infections, such as pneumonia or shingles, and to provide special protection in people with certain medical conditions. And as you're seeing with COVID, it's expected that everybody should get that vaccine. And here you've seen this in the last unit. There was a review sheet that went over the childhood vaccinations, but this just sort of summarizes that up again. Okay, that is it.